So we remember that polar things are soluble in polar things, and nonpolar things are soluble in nonpolar things. And so we are going to exclude the nonpolar things away from the water. Remember, that's the hydrophobic interactions. We don't want that very organized water sort of cage around anything that is hydrophobic. So what they're going to do is they're going to come together to minimize that. So what you end up having with these phospholipids is having, and this is not to scale. It's like having a person with a big head in a cartoon. This is not to scale. We're just trying to show you that this blue thing is the phosphate, and then these are the two. When you have a phospholipid, you have the two fatty acid chains, and on that third carbon we have that phosphate group. So here we have the phosphate groups on both sides of our bilayer, and then on the inside we have our fatty acids. Now you can see some of them are kinked, some of them are not. They're sort of floppy. Now, one important thing about the lipid bilayer that is we can, because this interaction keeping these guys together is very weak, that we can have an action going right to left of these. So see this guy, this one, and say this one? They're not always going to be opposite each other. They don't stay static. So they'll be moving, so the two layers kind of go like this. They move past each other. However, say this was on the outside of your cell, and this on the inside, they're not going to flip-flop. They're not going to flip-flop. So here you have a blow-up of a cell here. The blue parts are phosphates, and the little lines, not to scale, are fatty acid tails. They're going to be in there. And then on the inside, we have the phosphate groups. Now, and I believe I have a video of this where we can see that this part, the inside, can almost like turn. It's almost like you have the inside that can turn and go. So this is the outside layer. This is the inside layer of my phospholipids. If you're inside, you're always going to be inside. If you're outside, you're always going to be outside. But they can move past each other. It's kind of cool. Now, why is the lipid bilayer so awesome? Because if you're going to get through, you have to be both very polar and very nonpolar. How many molecules do you know that are both very polar and very nonpolar? Not really many things can do that. So that's why we're going to do stuff to this bilayer to let things through. Small molecules, actually, gases can get through oxygen. Yes, Alex?
How about shuttles?
Yeah, our shuttles, that's what we're talking about. There might be actually pores to let those molecules through, but we'll see about that in a second. So I mentioned that the inner layer and the outer layer float past each other, but they're not created equal. They can have different stuff going on. Here's our little key. This is a cerebroside, so you've got that going on here. The basic idea of having something polar on the outside and nonpolar on the inside is really the basis. But then you can have lots of stuff going on. Every once in a while, we're going to throw in a cholesterol to be that little piece of cardboard to add some rigidity. We generally have the bulky ones on the outside. Why would we have the bulky ones on the outside? Stuff with lots going on. The ganglioside, the cerebroside. Why would you want it on the outside?
So it can interact with itself?
Yeah, so it can interact with other things in the environment. Especially if you have sugars, you'll see that it's detecting things in the environment. So it's held together by very forces. I find it ironic that one of the most important things, or the most important thing for life, depends on the weakest forces out there. All right? The weakest forces, intermolecular forces, are van der Waals and hydrophobic forces. And what's necessary for life, the cell membrane, is held together by those weakest forces. But it works, or we wouldn't be here. All right, so that surface is going to be charged and has crazy stuff going on. So in our lipid bilayer, let's talk about the fatty acids that can be inside. It can be saturated or unsaturated. And by having the unsaturated, it's going to make it more oily, more flexible. So if we have lots of saturated, it's going to be close, uniform packing. And that unsaturated provides kink, more space, more fluidity. And if you have more unsaturated, it's going to be a more oily bilayer. And you can see plants will contain more unsaturation in their bilayers. We can characterize a lipid bilayer by finding out when it becomes disordered and just really just melts. And it's a characteristic temperature. So we describe, okay, this cell or this kind of membrane has this TM. So if you have lots of unsaturated fatty acid, it's going to decrease. I'm sorry, it's going to decrease. This is a typo, the TM. So if we have the cholesterol in there, you'll still get this disorder, but it won't lead to an increase in fluidity because it's a piece of cardboard adding the rigidity. You have a question?
No, I'm just looking. Sorry.
Okay. All right. It looked like you were not understanding, like I said something wrong. All right. So if you add in a cholesterol right here, cholesterol isn't going to change. It's not going to melt, right? The ring structure is rigid no matter what temperature it's at. So say we have cholesterol in here, and even though these guys are sort of melting and becoming more fluid, adding in a cholesterol every so often is going to lead to almost a permanent rigidity to your membrane. All right, so in our membranes, we can add proteins. And there's different types of proteins. They can either be peripheral, hard to say, or integral. So integral proteins span the lipid bilayer, and peripheral ones are just adhered to the top. So what can we say about the peripheral protein as compared to the integral? What's on the outside? What kind of amino acids would be on the outside of our peripheral? Remember our 4 classes of amino acids? Nonpolar, polar, acidic, and basic, right?
Polar.
We put more polar ones. How about our integral? We would have nonpolar. And so really, in our integral proteins, they're nonpolar on the outside so that they can stay here inside the nonpolar part of our membrane. Sometimes we see these integral proteins as sort of inside-out proteins, where the polar part will be in what we call a pore, right, to allow polar things through the membrane. And then the outside will be nonpolar, so that it can interact with the nonpolar part of our membrane. If I had to do a project like this, studying membrane proteins in graduate school, I probably would have picked an integral protein, because once you try to study it, it becomes denatured or inactive. And I had a knack for picking the most difficult projects that would result in failure and wasted months and years of my life. Why take the easy road? This is crazy stuff. Look at this. This is an example of a membrane protein that would span it, often alpha helix or beta sheet. that would minimize the polar to nonpolar interaction. So wherever it's polar is going to be away from the membrane, and wherever it's nonpolar, it's going to be close to the membrane. Sometimes you'll see a lipid anchor where we have a cysteine residue that will interact with a fatty acid. Cool.
Can you say that again?
You can have an anchor that will kind of sort of keep our protein close to a certain fatty acid. And so what you're doing is you're having this chain. The blue is the chain of amino acids. And here's a cysteine amino acid with a sulfur. And that, remember how sulfurs can nucleophilically attack the carbon of a carbonyl? So here we have a covalent bond between an amino acid in this membrane protein and a fatty acid that's part of our membrane. We can have other things going on as well. And we can have, I'm not going to talk about transport proteins yet, but we're going to talk about membrane transport later on. But we can have transport proteins that allow substances in and out, or receptor that bind to signal molecules that will tell the cell what's in the environment. We can have enzymes attached to the membrane, either inside or out. But it's a crazy sort of look to the membrane. All right, we introduced it as this nice little neat thing of phospholipids, right? But really, what it looks like is this. Not really cartoony, but there's crazy stuff going on. So here we have integral proteins. They look differently. Here's something with a alpha helix. Here's something more of a channel. These green things are going to be sugars. Here we have cholesterol, sort of put different places to add that rigidity. Glycolipids to signal. So anytime you see sugar, you're going to think, oh, we're detecting what's in the environment. Look at that. Is that crazy? So again, the peripheral proteins are loosely attached, but the membrane, the integral proteins are in there. And oftentimes you'll see them covalently attached. And this one works.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Is that crazy or what? It's crazy that 2 molecules thick is what life depends upon. Now, I want to mention before we leave this is that, so this is our cell membrane, and the difference between eukaryotic and prokaryotic cells is that sometimes the bacterial cells will have this cell wall on the outside that will give it more toughness. Remember the cross-linked cell wall. So when you work with mammalian cells, they're very easy to break apart. But when you work with prokaryotic cells, they're a lot tougher to break apart because they have additions on top of this. And I'm going to end